Okay, so now that we understand what Mendel was doing, and he was basically taking the gametes from a male plant and fertilizing them into the gametes of a female plant to make a zygote, which would then grow into a new seed that he could put in the ground and get the babies and grow them, he started to realize that he could compare things. And so another scientist named Punnett, at a somewhat similar time, although my understanding is it was a little bit after Mendel, started to realize that you could make it something that would easily allow you to figure out what the probability was of certain babies having two. So he did things where he took moms and he crossed them with dads. So let's talk about the height gene. So that means you can either be big T or little t. And we'll take a mom who is homozygous recessive. So homo means same, and recessive would be the little. So she has two little t's. And then we'll take a dude who is heterozygous. So he's little t, big T. So what Punnett figured out is that you could put the different gametes. So notice that this mom can have a gamete that's little t or a gamete that's little t here. That would be the egg. So those would be two different types of eggs that she can make. And then this male, because he's heterozygous, can make two different types of sperm, one that has the big T and one that has the little t. So if we put the mom on the top of the Punnett square, we know the mom can be little t or little t, so the only two gametes she can be are those. So you always put the gametes here on the side, gametes, and so the gametes are also going to go here. Now for the dude, the gamete can be a big T or a little t, so we'll put the big T there and the little t there. Those are the two possible gametes that the male can make because of his genotype, and these are the two possible gametes that the female can make because of her homozygous recessive genotype. Now if we want to figure out what kind of babies then they can make, each of these squares would represent a different baby that was made from a different fertilization. If we fertilize the sperm of this guy into her, then it would be big T, little t. This one would also be big T, little t. And this one would be little t, little t. And then finally, if this sperm went into this egg, that would also be little t, little t. These are the genotypes. But what we ultimately want to know are the phenotypes. What does the plant actually look like? Since this would win, this one would be tall. This one would be tall. This one's recessive, so it's short. And this one is also short. And Mendel would have identified that if you do a homozygous recessive female with a heterozygous male, you have a 50% chance of being short and a 50% chance of being tall. And that was pretty spectacular, and he did a bunch of those. But what he really wanted to know was this. If you have the height gene, and you also have the seed color gene, do they go together? In other words, is a tall plant always green, and a short plant always yellow? Or can they swap and go with a different genotype? This is called the law of segregation. He was trying to prove, do the genes always go together, or do you get a total mix match of all the genes? And so he did something frickin' brilliant. First off, he took two parents so that he could make some seeds that were looking at two genes at a time. So the Punnett square we did before was a one-gene Punnett square. But what we really want to do is a two-gene Punnett square, because then we can see, did the genes go together, or did they mix up and go in all sorts of different funky ways? And then we would be able to figure out, which is what Mendel wanted to know, is do genes go together or do they segregate independently? You already know the answer because you took the quiz, that genes independently segregate. But he has with the guy who proved it. Here's how he did it. So he took two parents, a male and a female, that were both heterozygous for two different genes. So we're going to talk about the height gene, and we're going to talk about seed color. Height can be tall or short, and seed color can be yellow, which is dominant, or little y, which is green. So little y is recessive, green, and big Y is yellow, it's dominant. So we took a female that was big T, little t, and big Y, little y, and crossed it with a male that was the same way. So that he basically set it up with every possible gamete you could make. And he wanted to see if I could make every single possible baby possible. Would they all look the same? Would there only be 2 types? Or would there be a whole boatload of types? If there was a whole boatload of types, he knew that the genes independently sorted. He then went on to grow literally thousands and thousands and thousands and thousands of pea plants, because that was the only way he was going to get enough that he could prove whether he was seeing lots of different phenotypes or whether there was just a couple and they weren't segregating. So basically he took a female who was T, T, Y, little y, and a male who was also big T, big Y, little y, and he mated them together. So these are the hardest ones, because what we have to do is figure out what are the gametes. So we need to figure out here what are the possible gametes, which means we have to go and match each one of these with each other. I always do this remembering FOIL. Remember from math when you learn FOIL, which says this one goes with this one, this one goes with this one, this one goes with this one, this one goes with this one? It's exactly the same. This one will go with this one, this one will go with this one, this one will go with this one, and this one will go with this one to get the 4 possible gametes. So the first one is big T with big Y. Big T with big Y. The next one is big T with little y. Big T with little y. The next one is little t with big Y. Little p, t, big Y. And then little t with little y. So he was trying to mate where he would get every different possible gamete possibility. And that what meant if tall always went with yellow, then the only kind of babies he were going to get were tall yellow ones or short green ones. But if he got every possibility, then he knew that the genes went separate from each other and that you could get every possibility possible, and this would create great diversity in all of the different plants. You already know the answer. There is great diversity, and you do independently sort. You would have to do the same FOIL here, and you would get the same things. Big T, little y, big T, big Y, big T, little t, big Y, and little t, little y. Then you have to cross every one of these. So I'm just going to do a couple. This one will be big T, big Y, this big Y. This one would be big T, little y. This one here would be little t, and big Y, big Y, big Y, big Y. And this one down here would be little t, little y. And you could fill in all of them, which is what you should do to practice, although I know you had the test already. So this would be the genotype for each of the different zygotes that you would get from some of these sets of gametes. And now we can figure out what the phenotype is. Well, this one would be tall. This one would also be tall. This one would be short. This one would be short. This one would be yellow. This one would be green. This one would be yellow. And this one would be green. And by doing this thousands of times, Mendel showed that sure enough, the genes independently sort, and we get every possibility of the gametes, which leads to great diversity. And even when you have many, many, many genes instead of just 2, they will independently sort. And that gets us up to date on Mendel, and later on we'll learn about other types of genetics.